Hello everyone. So today we are going to discuss about the bacterial cytoplasm. Bacterial cytoplasm here, basically it is a dense, thick, uh, fluid-like structure. It is a dense, thick fluid which is found within the bacterial cell. So that is basically the major component in which the entire bacterial cell is consisted upon. Previously, we were done with the outer coverings of bacteria, which included the bacterial cell wall, bacterial cell membrane, as well as we had an insight into the glycocalyx as well. But now we are going to discuss about bacterial cytoplasm, which is all based upon some of the components which we already discussed with in our previous chapters. So here in bacterial cytoplasm, there are no membrane-bound organelles present as obvious, but there is, ribosome, as it is non-membranous organelle, and here ribosome basically is smaller ribosome, smaller in size, 70s, and it has two subunits: smaller subunit, larger subunit. Smaller subunit is 30s and larger subunit is 50s. While when it comes to the ribosome, well, when it comes to its number, it's found huge in quantity, as well as some of the ribosomes are also attached to the plasma membrane. And apart from that, these ribosomes are only responsible for intracellular protein synthesis. Apart from which, when it comes to some other characteristics, such as some other organelles, we are have not having any particular membrane-bound organelle, but around the plasma membrane, we are having certain invaginations. Just like if we talk about these invaginations, here we have this plasma membrane, and here that plasma membrane is a bit invaginated inside, and this invagination of the plasma membrane inside the bacterial cytoplasm causes to form several infoldings or invaginations, and these infoldings or invaginations are known as the mesosome. And these mesosome are of two types. First includes the lateral mesosome, and second is the septal mesosome. When it comes to the septal mesosome is the one which basically involves the septal mesosome is the one which basically involves uh, replication, DNA replication. As we know that mesosome is associated with respiration as well as DNA replication as well as some other sort of functions. We are going to, with the types such as septal mesosome is associated with replication, such that it is a site where the plasma membrane is associated with DNA replication, and lateral mesosome is the one which is containing the respiratory enzyme, or is having the plasma membrane's portion which acts as a respiratory enzyme, due to which it basically is the site where the plasma membrane is associated with respiration. Apart from which, there are several vesicles for photoautotrophs, such as photoautotrophs have several vesicles near the plasma membrane in the cytoplasm. And these vesicles near the plasma membrane in the cytoplasm, which contain the photosynthetic pigment, bacterial chlorophyll, and chlorobium, are basically known as the chromatophores. Chromatophores. These chromatophores are only found in photoautotrophs except cyanobacteria. Cyanobacteria do not have chromatophores, but they do have something else, which we have previously discussed, known as the thylakoid membrane, right? And apart from that, when it comes to some other stuff, here we are having the nucleoid material, the genomic material is also dispersed within the cytoplasm. There is no specific organelle in which the genetic material is present. There is, the specific uh, cytoplasm, or the region of cytoplasm in which the genetic material is present, known as nucleoid region. Apart from which, there are certain extra chromosomal material is also present, which is known as extra chromosomal material is also present. Extra chromosome material is also present. So just small circular extra chromosomal material is also present. Extra chromosomal material is also present. This is what do we call it? The extra chromosomal material, which is known as the pillars. Here we are having actually we are having extra chromosomal material, which is known as plus. And here, within the next video, we are going to discuss about the bacterial appendages.